Ascidian has been a wonderful partner of the Carroll Center for the Blind, including serving as a sponsor for our annual Walk for Independence. They also provided an internship to one of our Real World of Work experience students this summer at their office. And they are now underwriting this three-part educational series, which began with 2 webinars in the spring and summer, and now we're wrapping up with this wonderful in-person event. So I'm honored to have 3 members of Ascidian here today. We have Robert, aka Bob Bell, Chief Scientific Officer, Aliyah Rashid, Vice President of Clinical Development, and Amber Trezak, Patient Advocacy and Engagement. So without further ado, I'd like to invite Bob Bell up here to give us an overview of Ascidian. Thank you.
Thank you. Good morning, everybody. How are you? It's a real pleasure to be here. As mentioned, I'm Bob Bell, CSO of Ascidian. I am about 5'10", 5'11" on a good day. I have dark hair, and I'm wearing glasses today. And you know, Ascidian was founded with the concept of rewriting RNA for the treatment of human diseases. And can you not hear it well? You want me to speak louder? Yes. Okay, great. I'll try to get a little bit closer to the microphone. Is that better? Okay, good. So Ascidian was founded with this concept of rewriting RNA. I'm going to talk a lot more about that in a later session of what that actually means. But to do that for the treatment of genetic disorders such as Stargardt's disease, which is our lead clinical program. So Ascidian is a Boston-based biotech company. We're a clinical stage organization. We're about 3 miles from here, actually, our laboratory space in the Brighton area. And the company is just under 50 people today. So we've been pretty lean in terms of taking our first program to the clinic, as well as developing a pipeline of therapies for other projects. The company has a fantastic leadership team with decades of experience in drug development, both in large pharma as well as in small biotech, and in particular in gene therapy, which is the approach that we're taking. Most of the company is made up of a group of amazing molecular biology scientists that every day are working hard in the lab, as well as complemented with an amazing operational team that makes sure everything we're working on is up and running, as well as a great clinical team, for which Aliyah is here today and can talk a little bit more about our clinical program. So the company was founded in March of 2020, so right at the onset of COVID. So we consider ourselves sort of a COVID baby company, which had its own challenges. And essentially, we took inspiration from nature. That's actually where the name Ascidian comes from. So Ascidian is the scientific term for sea squirts, right? The little ocean creatures. And they do an amazing thing with their RNA that, again, was sort of the inspiration behind the technology that we're working on. In January of this year, we opened our first IND. That's an investigative new drug application with the FDA to start a phase 1/2 clinical trial in Stargardt's. So a really great accomplishment from going from 2020 to this year and already being in the clinic. And then another exciting event earlier this year in the summer, we announced a partnership with a Swiss pharmaceutical company, Roche, on a setup, not in the ophthalmology space. In particular, we're working on neurological disorders with them, but that was a really exciting piece. And as mentioned, our clinical program called Stellar is actively enrolling and off and running. So the main things that we were thinking about when we set up Ascidian was, how could we leapfrog current genetic medicine technology? Where could we tackle problems that existing approaches, existing modalities could not address? And so we had a few sort of fundamental ideas behind that. Number one, we wanted to edit RNA and not DNA. And I'll get into why that is a little bit later on. We wanted to be able to make very big changes, not just single edits at a time. So we think about it, instead of changing a single letter in a book, we can rewrite entire chapters. And that's really important because it allows us to address a much bigger population of the Stargardt community with a single medicine. We did not want to use any foreign enzymes. Folks in the room may have heard of things like CRISPR-Cas technology that essentially is using bacterial or synthetically derived proteins and enzymes to allow for their editing reactions. And that can carry its own challenges in drug development. These bacterial enzymes can often be seen by the immune system as foreign, which they are, and it can also be very difficult to actually deliver these types of technologies to the cell. So we wanted to avoid any type of foreign enzyme. We also wanted to be agnostic to delivery vehicles, and what we really mean by that, we wanted to make sure that we could deliver our RNA exon editing technology with clinically proven approaches. We didn't want to have to innovate on top of innovation when it came to delivery. And so as you'll hear a little bit later, we're using a very standard approach and adeno-associated viral vector that has a lot of clinical precedent for the ACDN-01 program. And lastly, we wanted to make sure that we could maintain the normal gene expression of the targets that we're going after. In genetic medicine, you want to make sure you're getting correction in the right cells, but also at the right levels. And as I'll talk about in a little bit, the RNA exon editing approach covers all of those things. So our key take-homes, right? We edit RNA, not DNA. We do it at a very large scale. We don't have to use any foreign enzymes. We can use clinically validated delivery methods, and we can ensure that we're maintaining the expression in the right cells at the right time. We're really excited about the Stargardt program, but also building a pipeline in other areas such as ophthalmology, neurological and neuromuscular, and other areas of genetic diseases. We think the technology has a lot of applications, and again, really excited to initially prove the benefit in Stargardt's. So that's all I have just for a quick introduction. What we're going to do next is Amber is going to talk a little bit more about just the drug development process, what different clinical stages actually mean. I'm going to come back up and talk a little bit more about the science, this RNA splicing that I mentioned, and a little bit more about the data for the lead program that we provided to the FDA to enter into a phase 1 clinical testing. And then Aaliyah will come up and talk about the clinical study itself. And then at the end, I think we'll have plenty of time for questions after that. So again, thank you all for having us here today, and we look forward to continuing on the discussion.
Okay. Can everybody hear me okay? I talk quite loudly, so just tell me like lower if I am too loud. So thank you guys so much for being here. Being a part of Ascidian, we're super excited about our partnership with the Carroll Center, and for everybody in person today, for us to provide this opportunity to you all to speak to what I'm going to talk about, the ins and outs of clinical trials before we dive into the scientific and clinical aspect of it. So I'm not sure how many of you remember the 80s and 90s, the more you know, with a little star, and it's like, doo, 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 you know, that platform for PSAs. I think that's such a valuable phrase and kind of my motto for this platform on educating folks about the ins and outs of clinical trials, and more importantly, the history of the drug development process. And that way you can be a valuable participant, not just maybe volunteering for a clinical trial, but also understanding how you get to that point. And that's just really important. So for me, it's the more you know, the more you can understand and be an active part of this information. So I'm going to start with some statistics that are a little bit frightening and scary and pretty big. And just, again, the more you know, on average, it takes around 10 to 15 years and costs about $2.6 billion to develop one new medicine. That is from concept to production. So that's a pretty darn long process and super duper expensive. In the world, there are around 7,000 known rare diseases, but only 5% of those diseases actually have a treatment on the market. So there is definitely work to be done, and Ascidian's doing that work in the Stargardt community. So To get started, you kind of have to start at the beginning to understand the drug development process. And there's this overarching entity called the FDA, so the Food and Drug Administration. That's the organization that rules and provides all of these guidelines and restrictions and criteria that drug developers, also known as sponsors, have to follow. So the FDA is the organization that we adhere to, and they're the people that review all of this information and data on drug development and allow you to either proceed, not proceed. They give you guidance, so they are very important. So where do you start? Drugs or devices, but in this case, we're gonna start with drugs. That's what I'm gonna mainly speak to. They always start as a concept. So somebody perhaps is reading literature on, a disease or a condition, and they come up with this idea. And this could be a single person, like a parent, or it could be a 2 or 3 person lab. As Bob spoke, Ascidian started in 2020 with a group of folks that kind of decided to go down the Stargardt route. And it could also be a part of a bigger pharma. This phase is called the discovery phase. So this is where all drugs and devices start. They get their brainchild, so to speak. Once you move out of this phase, these researchers or scientists start to kind of create these hypotheses, and they start to develop these concepts and start to bring them to action. So these are still just more or less thoughts and little minor experiments that they're doing in the labs. And sometimes this can take years. There's a lot of trial and error at this point, a lot of research, a lot of reading. These guys are super duper smart. And this is usually referred to as the R&D or research and development phase. And so once they have something that they think is going to stick and could work out, they start moving it into preclinical research. This is where researchers start to take their concept, their hypotheses, their ideas, and they move them into testing. They test in vitro, which means in cells, so little petri dishes or vials. And then they also move into testing in vivo, which means in animals. So they're taking this concept of a drug or a treatment and testing it against these models that are in these cell dishes or in animal models. And animal models can be anything. You can use mice, you can use rats, you can use dogs, all sorts of things. But those guidelines are set by the FDA as to what makes most sense to do this preclinical research. The main purpose of preclinical research is to make sure that the researchers or the drug developers are able to determine the amount of drug that is considered safe and that it's non-toxic. So once they do all these experiments and they gather all this data, if they feel it's promising, they will then submit, as Bob said, an IND, so an investigational new drug application, and this gets submitted to the FDA. So it's like a book, right, that has all of this information, all of this data, and they submit it to the FDA. And not only does it include all of this R&D data that has been collected up until this point, which, mind you, could potentially have been 5 or 7 years at this point. So if you think about the grand scheme of things and, kind of putting a pause in it, you look back to, there was these 2 scientists that had this idea and they start testing their hypotheses, and then they test it in petri dishes, and then they test it in animals, and they find that it's finally something worthwhile, something that they think will change the lives of people. They fill out all this application. They come up with a protocol, which is called in layman's terms, a clinical study design. It's a plan. It's a plan that tells you step by step what this drug testing trial should look like. So it includes assessments. It includes who's allowed to be in the trial, who isn't allowed to be in the trial. It includes how long the trial is going to last. So this package, this IND package that is submitted to the FDA is extremely intense. It's very labor intensive because it's not just like, oh, hey, we think it's going to work. It's legit information. And so the FDA will then receive this, and they're going to start looking through it. So once they receive this IND, the FDA has 30 days to acknowledge the receipt and either come back with, nope, that's not good. And those researchers that started perhaps 5 to 7 years ago have to start back at the beginning again. Or the FDA could say, you know what, we have a little bit more questions. Maybe could you run an additional study? Maybe additional toxicology because it was a little confusing. So can you test it a little bit more? Or perhaps they're like, nope, this looks great. We agree with you guys. Now you can move into clinical research. So you've done the research and discovery phase. You've done preclinical research. Now you're moving into clinical research. This is the first time where this drug or device will reach people. So up until this point, it has never been tested in people. So this is a very important phase, and it's very valuable for people to be well informed and to understand that clinical research isn't just overnight. There is a legacy of information that the FDA has reviewed to say, I'm giving you guys the green lights as sponsors to start pursuing this treatment in people. So clinical research, excuse me, once the FDA gives the little green stamp of approval, you're starting this thought process of clinical research. Once an IND is accepted, as Bob said for Ascidian, that was in January. That doesn't mean magically patients start getting treatment. Unfortunately, there's a lot of back-end work that goes into it. So if you remember, I had mentioned there is a protocol as part of this package. This study design plan is what investigators or the study doctors who are running the clinical trial on behalf of the sponsors have to follow to a T. So these sponsors, these drug investigator or drug developers, work with these investigators to make sure that their trial site is set up, that they understand how this drug is administered, how people are included or excluded, the reasoning behind it. So all of this kind of legwork goes into it once an IND is accepted. Then you can start enrolling people into these clinical trials. So speaking of clinical research and clinical trials, there are a lot of types of clinical trials out there. And I feel like most people are familiar with clinical trials. They range in numbers. And so sometimes folks understand like, oh, I've heard of a phase 1 or a phase 2. But I'm going to kind of break that information down for you guys and hopefully provide context so that when you do see these or hear somebody speak to them, you can say, I know exactly what that means. So there are technically 5 clinical trial phases accepted by the FDA or acknowledged by the FDA, and they're sequential, which makes it super easy. But as with most things, it's not just black and white. So a clinical trial can be considered a phase 1-2, which is what Ascidian's is, and Aaliyah will speak to that a little bit later on. It could be a phase 2A, 2B. There's all these, nuances that go into these clinical trial phases, but I'll try to keep it as simplistic as possible so that I don't overwhelm you guys with all of this information. But it starts with a phase 0 clinical trial. And not a lot of people actually have heard of that, just probably because it's non-interventional, which means it's not testing a drug. It's usually just a registry or a data bank, a collection of information for sponsors, drug developers to store, to get a bit of an insight into that community or that population. So that's considered a phase 0. It's non-interventional. It moves then into phase 1. You guessed it, right? And this is where most sponsors start. Most companies start with a phase 1. So I'm going to speak a little bit categorizing these trials to different categories. So one is like an oral medication or infusion or an injection. And for those types of phase 1 trials, they traditionally start in healthy volunteers. So that means that participants do not actually have the disease or condition that the drug is intended to treat, which some people are like, why the heck would you do that? Well, the intent of a phase 1 clinical trial is first and foremost safety. That is the most important thing that drug developers and the FDA are looking for. So there are people that volunteer to participate in clinical trials for a disease or condition that they may not have. And that's a great benchmark to determine, are there any adverse side effects? Are there any tolerance issues? Should we, basically look at the different design? Should we give it twice a day versus once a day? So the whole impact is to look at all of these side effects. In contrast, in gene therapy trials, you actually start with participants that have the disease or condition that you're trying to treat. You don't start in healthy volunteers. So phase 1 trials in gene therapy always include participants that have that disease or condition. And so this is exactly what ACDN-01 with our Stellar trial is. We are enrolling patients that actually have Stargardt disease because we are considered a gene therapy trial. After all of the data and information is collected after each phase, it always gets submitted back to the FDA. There's always the opportunity to have an open dialogue with this organization. They always want to know what's going on. You have the right to request meetings as a sponsor, to ask questions. And if the information submitted to the FDA after your phase 1 clinical trial is deemed good and positive, they allow you to move on to a phase 2 clinical trial. So typically, phase 2 clinical trials are a little bit larger. They include more participants. They include everybody that has the disease or condition that you are trying to treat. So that's the vast difference between a phase 1 and phase 2. Phase 1s can have healthy volunteers. Phase 2s always include those that have the disease or condition that you are trying to treat. The main goal or purpose of a phase 2 clinical trial is to test the efficacy, or does it actually work, of the drug or device that you are testing in these participants that have this disease or condition. And again, also, what are the side effects? That's always monitored. That is of the utmost importance. Those are never overlooked. And it can be simple side effects, such as maybe a rash or a headache, nausea, anything that you see on medicines that you take. Those have all been captured in a clinical trial at some point. The other difference in phase 2 clinical trials is they can be classified a little bit differently. So they can be considered open label or placebo controlled. So open label means everybody gets the drug. So it doesn't matter who is enrolled in the trial, everybody is getting the drug. And of note, Ascidian's clinical trial Stellar is this example. We are open labeled. The other contrast to that is called placebo controlled. And this is where you have a set of participants that don't actually get the drug. They get an imitation version of it that is placebo. So if your drug is a capsule, they look exactly the same. And the participants don't know if they are having the actual drug or if they're having a placebo-controlled option. And if you do the placebo-controlled route, you can do unblinded or blinded. So blinded means nobody knows who gets the drug, nobody knows who gets the placebo. And the reason you do that is some scientific reasoning of bias. So you can control the effects of that. You're not being biased that folks have this thing called, placebo mindset, that they may think that they have that. But, they can also make it unblinded. Excuse me. And that's where everybody, who has that. Thank you. Excuse me 1 second. It's going around. So if a phase 2 trial is considered to be successful, the FDA then approves you to go on to a phase 3. But at this point, I'm going to throw another statistic out because, again, the more you know, at this point, only 33% of drugs move into a phase 3 clinical trial. So that means, my math is terrible, on average, 70% fail. So they fail for some reason. Maybe they're not showing efficacy. Maybe they have too many side effects. Maybe it's actually not working. So 33% are only successful, which is really scary because if you think about it, numbers are just dwindling and dwindling and dwindling, and it's getting harder and more challenging. And so that just kind of advocates that we still need all this research to get out there to make drugs successful. So moving on to phase 3 clinical trials. This is to determine if a drug or device offers treatment for a specific disease or condition within the widespread population safely. The main information gathered from a phase 3 is safety, shockingly, which determines long-term side effects. So in comparison to a phase 1 or phase 2, a phase 3 often is longer term because you're doing it now over a longer period of time. It's in a more broad community or population of those that have that disease or condition. And it's often the largest version of a clinical trial. To be considered successful in this phase, this is the FDA's requirement, is that this drug must be considered safe and prove to be more effective or as effective as current treatments on the market. So why this is important to kind of digest is that drug developers can create all of these opportunities for members that have a disease or condition, but if it just barely meets what a current drug on the market is doing, the FDA is like, not worth it. So therefore they decline your application and you're unable to proceed. However, once your trial for a phase 3 starts wrapping up and sponsors say, gosh, I think we have something, this is promising, you're getting these data report outs, they can seek approval from the FDA by submitting another package that has a different name. So this is an NDA. It is a new drug application. So before, when you submitted the IND, mainly it was your preclinical research. Now for your NDA, not only do you have to include all of your preclinical research, you have to now include all of your clinical research that you've captured up until this point. So this includes data that you've received from people. So testing your drug or device in people. All of this information goes into this NDA. Now, this is where it gets a little frustrating at times. The FDA has 6 to 10 months to review this NDA package. So that's like, whoa, right? They don't even communicate. You submit it, they have 6 to 10 months to respond back to you. However, if the sponsors determine that their drug is meeting an unmet need or there's no other drugs out on the market, they can appeal to have it called fast-tracked. And that means the FDA has to review it within 6 months. So it kind of cuts the timeline a little bit in half. I think that those timelines are very important because as we advocate to try and get drugs to the market faster, there are some limitations and you can only move as fast as you can, right? So I think being aware and cognizant of these timelines is helpful in understanding that it's not necessarily entirely on the drug developers. It's also not necessarily on the FDA because they're getting hundreds of applications all the time because there are so many diseases out there. So another benefit is if you go the fast track route and your data is super promising and it's deemed really good and approved by the FDA, it can be elevated to accelerated approval. So this is something we saw a little bit with the COVID vaccines in 2020, 2021. So they can grant authorization based upon the evidence at the time that was presented. And again, they will only consider these designations for drugs that have a new or better treatment option for a disease or condition that currently exists. Wrapping up with our clinical trial phases, there is a phase 4 clinical trial, and this happens once a drug has been on the market. So this is a very long-term study to collect any sort of information that may be missed. So the drugs are on the market, it's being produced quite largely, it's being prescribed quite largely, and it's the FDA's opportunity to, in real time, keep its fingers to the pulse of this drug or compound that was approved. It also occurs without being in these restrictive settings of a research facility, and it can provide feedback on information that may not have been noticed. So drugs that you typically see commercials for that have the catchy like jingles, and I'm just thinking off the top of my head that they have all those phrases and jingles. Those drugs are currently in a phase 4 clinical trial. And so what it is once it gets to market and folks start testing it and using it, if you have a side effect, you can report it to the government and they will then track it. And that kind of goes into this whole data package. So those are the 5 phases of clinical trials with a little kind of background information on the drug development process. And I just kind of wanted to wrap up with a little bit more statistics, that it's super hard for drugs to actually get through all phases of clinical trials. And it's estimated that 85% of drugs fail before reaching people. So 85% of drugs fail before reaching the market. So they fail in a phase 1, a phase 2, phase 3. At some point, 85% of drugs fail. So when you hear those astronomical statistics, the fact that we have drugs on the market now to treat, acid reflux just blows my mind, because there are so many smart researchers and scientists out there that are trying their hardest, in particular with rare diseases as well. So finally, in closing, some really exciting topics are, well, gosh, how many drugs are out there? How many are approved? And in 2023, we had the most drugs approved in over a decade. And we had 55 new drugs approved last year, which is pretty darn good, considering 85% of those were set to fail. And of those 55, 7 were considered a cell and gene therapy, which is pretty darn exciting things, because that means technology is advancing. In comparison, in 2022, we only had 37 drugs approved, and currently in 2024, we are at 38. So we're not going to hit 55, but we're getting there pretty darn close. And then lastly, I just wanted to share resources with you all, some of the organizations where I got my data and information from. So FDA.gov, they have a fantastic patient services department. You just do FDA.gov, type in patients, and they have a whole section. They even do videos on the clinical trial process to capture what I just covered. The Tufts Center for the Study of Drug Development, so Tufts University right here in Boston, has an entire research department ripping apart and investigating and researching different data around drug development, the processes, the costs, the timelines. Pharma, which is spelled PHRMA.org, is another great organization that works with sponsors to identify the timing around drug development process and costs associated with it. And then the clinicaltrials.gov, that's your main resource of any regulated FDA organized registered clinical trial. So clinicaltrials.gov, you can search by condition, you can search by disease, you can search by intervention. It's a great resource to review all of those. And then lastly, the Ascidian website. So we're just Ascidian-tx.com. And on our website, we have a page focused on patients and care partners. And we try to make all of our information as lay friendly as possible. But we'll be here the rest of the morning if you guys have further questions, and save them for the Q&A. So thank you guys so much.
Okay, so now that we know how drug development works, I get to come back up and talk about science, which I love to do. And as a scientist, we really live in an amazing time of genetic medicines. We now have the ability to understand the genome, to understand our DNA and our RNA and our proteins, understand how mutations cause disease and where those mutations are specifically located in genes, allowing scientists like the ones at Ascidian to come up with novel therapies. And at Ascidian, we try to leverage very large human genetic data sets. We're taking advantage of very state-of-the-art tools in molecular biology. We have an amazing computational team that's starting to apply machine learning and artificial intelligence learning towards the development of drugs. So it's really an exciting time for genetic medicines and gene therapy. So you hear a lot about gene therapy, and that can mean a lot of different things. But at Ascidian, it means one fundamental thing, that we are going to try to correct the fundamental cause of the disease. If we can correct the gene that has a mistake in it, you now have an opportunity for radical efficacy in the clinic and for patients. And so that's the way we think about gene therapy. So as I mentioned earlier, the way that RNA editing technology at Ascidian works, it does not use any foreign enzymes. So how do we do this? Well, we're taking advantage of a process in every one of our cells called RNA splicing. Okay? So every one of our cells undergoes this process of RNA splicing. And the central dogma, I think folks have probably understood, is that you have DNA that's considered the code, that's turned into RNA, that's turned into protein. And splicing is a key part along the way. And I'm going to try a little bit of an analogy, a non-scientific analogy to help describe splicing. So imagine that you're a music producer, right? And you're writing a song, okay? And you need to edit that song for its final product. So you can think about the original recording from the artist as the DNA. It contains everything. It contains things that you want to keep in the song, things that you want to remove. So what does that music producer do? They make a copy. That copy is your first step of RNA, called pre-messenger RNA. So again, now that copy contains parts of the song that you don't need. We can call these introns. And then parts of the song that you want to keep. We call these exons. So it's that producer's job to remove the introns or the parts of the music that they don't want in the song and join together or splice together parts of the song that they want to keep. So bringing the exons together. This essentially is the same thing as RNA splicing. Our cells are these producers, and they're removing the parts of the song, the intron, that you no longer need, and joining these exons together. And that forms a mature RNA. That's the code for protein. So in this analogy, you could imagine, even in the parts you want to keep in the song, there may be some mistakes. Maybe there's a dog in the studio that barks during a critical piece. Maybe somebody accidentally knocks over a microphone, or somebody's instrument is out of tune, and you want to go in and correct those pieces, right, of the parts of the song you want to keep. And that's how Ascidian is thinking about our RNA editing technology. And instead of going in and re-recording one thing at a time and then having that producer make the change, what if we could replace half of the song all in one shot with one therapy? Much more efficient of a mechanism. And that's essentially what we're doing. We're able to rewrite RNA and change any errors, also known as mutations, that cause disease in the RNA. So let me talk a little bit about Stargardts and ABCA4, and hopefully this will all make sense. So ABCA4 is a very large gene. It's 50 exons, right? So the cell has to put all 50 exons together to form a functional ABCA4 protein. There are mutations in Stargardt's patients all across the gene. Thousands of mutations are known to cause disease. And so it makes it very difficult, again, to think about if you had a drug that could only correct one error at a time. So our lead program, ACDN-01, actually can correct exons 1 through 22. So essentially the first half of the ABCA4 gene in a single reaction. And this is all done, again, by using the splicing machinery within our cells. So ABCA4 is a really critical protein in the eye. It's expressed primarily in photoreceptors in our retina, and it's involved in recycling compounds that are required for our visual cycle process, right? So as we're perceiving light, it's turning it into a chemical reaction, and then that is being sent to our brain controlling vision. And ABCA4 is transporting some of these critical compounds for that visual cycle process. If the ABCA4 protein has a mutation and it's non-functional, what happens is these chemicals that are part of the visual cycle accumulate in the retina, in particular in the macula, which controls our central field of vision. Okay, and so what happens is these compounds, these lipids continue to accumulate and eventually become toxic to the cells in the eye. They become toxic to the retinal pigmented epithelial cells and then to the photoreceptors themselves, the light sensing cells in the retina. So, in theory, if we can restore ABCA4 function and protein levels in the retina, we can offer real opportunity to provide a life-changing treatment to patients with Stargardt's disease. So starting back in 2020, we decided we wanted to work on Stargardt's as our lead program. And we developed ACDN-01, which, as I mentioned, replaces the first 22 exons of the ABCA4 gene. So I'm going to talk now a little bit about how we did that and the data that we gave to the FDA in the IND application Amber was just mentioning. So one of the first things we had to do is figure out where can we get the ACDN-01 molecule to bind with the patient's ABCA4 gene. As I mentioned, we're correcting the first half. So again, to the song analogy, we want to replace the first half of the song, but the second half is perfectly fine. So patients who do not have mutations in that second half of the gene, we can actually use that ABCA4 RNA to help us restore protein function. And so we identified through screening thousands, actually hundreds of thousands of exon editor constructs to identify a set of lead candidates that we took into multiple preclinical testing. So what did some of those experiments look like? Well, first, we engineered cells in a petri dish to express ABCA4 RNA. It expressed normal ABCA4, but we also created versions where there were mutations, so mutations that would cause disease in patients. And then we could test the exon editors to ensure that we are precisely editing that RNA and restoring protein levels in those cells. And not only could we demonstrate that we restore the protein levels, but as I mentioned, ABCA4 is important for transporting these lipids and these visual cycle compounds. Well, we can indirectly ensure that the function of the ABCA4 protein is intact. So the edited protein that we generate still contains the functional activity to transport some of those molecules. So we spent a lot of time working in cells doing that and became quite confident in the efficiency of the reaction. But that's in an engineered cell line, not necessarily in photoreceptors. And while researchers all over the world are trying to develop models for growing photoreceptors in a dish, it really hasn't gotten to the point that it can be used for drug development, drug discovery. So we went directly to human retinas. We worked with an organization known as the Lion's Eye Institute. This is one of the leading centers for corneal transplant surgeries in the United States. And what's interesting is you can actually take these retinas from the donors who are giving their eyes for corneal transplant surgery, and you can grow the retina for up to a month in the laboratory. So that allowed us to test our exon editors, including ACDN-01, the clinical molecule, in human photoreceptors. We could treat these explants for up to a month and look at the RNA editing and look at the efficiency and also looking at the selectivity of that. So that was, I remember seeing some of that first data and a moment in my own career when I really felt like we were onto something. Demonstrating that we can edit the ABCA4 RNA in human photoreceptors was such a pivotal moment for this program. But that is still ex vivo, as we call it. It's outside of the body, right? So how can we be confident that we can do this in patients? And for that, we moved to animal models. In particular, we focused on doing experiments in non-human primates, okay? And so the way we deliver ACDN-01 is we use what's known as an adeno-associated viral vector. And this is now a very small amount of a solution that surgically, Leah can talk a little bit more about this, but surgically is delivered to the back of the eye. With a very thin needle and a very small amount of volume is delivered right to the macula to that region that controls the central field of vision that is in particular affected in Stargardt's patients. So then this AAV viral vector then starts to express the RNA exon editor, and it can go in and edit the ABCA4 RNA. So we dosed animals with different concentrations of the molecule, and we looked at different time points where we could take the retinas out and analyze the editing efficiency. We could analyze the editing specificity to make sure we're not editing other genes, for example. And most importantly, as Amber mentioned, to make sure we didn't see any signs of safety that would be concerning moving this into patient populations. So we looked, some of the time points we looked at 1 month after treatment, we looked at 3 months after treatment, and we've even gone out as long as 6 months after treatment and seen very durable editing out to 6 months. That gives us quite a high degree of confidence that the treatment effect for ACDN-01, we hope and think, can be lifelong. So a single injection allows for essentially lifelong persistent editing of the ABCA4 gene. And partially that's due to the fact that the photoreceptors in cells in the eye, they do not divide. So they're not going anywhere. So once you've delivered this AAV to those photoreceptors, it's going to be stable. And we have evidence from other gene therapy trials going out now even 10 years and seeing stable expression. So really a great opportunity for a lifelong, hopefully, treatment effect. So, and seeing that 6-month data and looking at the tolerability of the editing and the efficiency allowed us to select doses that we're now using in the phase 1/2 clinical trial that Leah will talk about. So we selected levels of the drug and the volume that we give that we believe will be well tolerated and safe and not associated with any potential serious adverse events in the eye. I mentioned a little bit throughout the way about selectivity. So with all gene editing technologies, you really want to make sure that you're editing ABCA4 and you're not editing other RNA in the gene. And so using those cellular models, the human retinal explant models, as well as all of these non-human primate studies, we did very deep non-biased sequencing looking at the entire what's called transcriptome. So essentially every RNA in the cell to make sure that we were not causing any effects on different genes or different pathways. And in fact, that is the case. We're quite confident in the specificity. And the low levels of what we call off-target editing that we do detect are orders of magnitude, in some cases 1,000-fold lower than mistakes that our cells make all the time. So again, giving us high confidence in the efficiency and in the selectivity of the drug. So with that data in hand, along with quite an extensive, what we call CMC or manufacturing process, we manufactured ACDN-01 with very high quality controls. We characterized it. We've looked at the stability of the drug, as well as making sure that it's a very clean product. And with that, we brought all of that information to the FDA along with the clinical plan and submitted that. And as Amber said, there's oftentimes a bit of a dialogue, I think about it as a collaboration with the FDA. It's not one side versus the others. I think throughout the process, we had multiple dialogues back and forth. They helped us think about the design of those non-human primate studies to make sure that we're really confirming the safety of the drug in these preclinical species. They gave us feedback on how we manufacture the drug and ensuring that the quality of the drug is going to meet their expectations. They gave feedback on the clinical plan and how we're going to monitor patients. And through that collaborative process, they cleared our IND earlier this year. And as Aaliyah will mention, we're now progressing the STELLAR trial. So we're really excited about ACDN-01. As I mentioned, the first drug is correcting exons 1 through 22 of the gene. And so we often get questions, well, what about mutations in the other part of the gene? That's something that we're working on. It's much earlier in discovery, but it is something that we're interested in and also pursuing. We're looking at other therapeutic areas, including other inherited retinal diseases, but also we believe that this RNA exon editing technology can be applied to many genetic disorders with significant unmet medical need. And so we're really excited to progress our pipeline both in the clinic and preclinically to really provide radical benefits to patients and families who are in need and have hope that we can deliver. So I think we can in the Q&A. Yeah. So with that, I think I can hand it over to Aaliyah to talk a little bit more about the clinical program. And then, as mentioned, we want to have plenty of time for questions. So if there are some, please save them for the next. Thanks.
I'm not as tall as Bob, so I'm just bringing the microphone in. Hello, everyone. My name is Aaliyah Rashid. I'm the Vice President of Clinical Development at Ascidian Therapeutics. So I'm a physician by training. I trained in ophthalmology, and within ophthalmology, I subspecialized in retinal diseases and the pathology of retinal diseases. So this is really, a place where I'm very happy, really enjoying what I'm doing, and feel very thankful to be part of such a great team working on something so important. So what I do in my role at Ascidian is take all of the great work that Bob and his team of researchers, scientists have done over the past 5, 7, 10 years, maybe longer, and help them to put together this great big book that Amber mentioned, this IND package, where we put together the preclinical data, and then myself and our Chief Medical Officer, Jay Barr, and the rest of the clinical team put together an argument for why we think that this drug will work in a particular disease. In this case, Stargardt disease. And, we send that off to the FDA. Thankfully, they gave us a thumbs up with no questions or concerns, and we're able to move forward into the clinic, which means we're able to test it for the first time in human beings. So we have a big task ahead of us. We have to write a protocol. We have to understand what the study design will be for taking this, this drug that has been developed and testing it safely in humans for the first time, and then taking that data, understanding it, and moving it through the development process. So hopefully we get to phase 3, and we're not one of the 85% statistic that don't make it. So, with my background, I've actually spent a lot of time in biotech working on gene therapies for retinal diseases, wet macular degeneration, dry macular degeneration, and other IRDs like Stargardt disease, so very rare retinal diseases. And, hopefully bringing all of that experience together, we can design something that helps us to get answers for safety, helps us to get answers for efficacy. So does the drug actually work? Is it stopping the disease? Is it maybe even improving some elements of the disease? And just moving it through the process as quickly as possible, because moving it quickly gives it a better chance of being a success and being approved at the end. And so what we're doing at Ascidian is we're actually doing a pre-screening study and a actual phase 1/2 clinical trial. So it's sort of, as Amber mentioned earlier, a phase 0, which is a pre-screening study, and then a phase 1/2, where we are able to test it in humans, make sure that it's safe, and then maybe test it in a larger group of people to really try to understand how well it works as well. The preclinical study, the pre-screening study is a really nice way of identifying patients, particularly when a disease is more rare in the population. So Stargardt is still a pretty rare disease. And often what you have to do when you're doing clinical trials is pick the best patients out of that population that has the disease that fits some characteristics that will help you to understand if the drug works. I think Amber alluded to this, but basically with gene therapies, you can't go into healthy volunteers for your first in-human studies. You have to go to a patient with the disease because it's a one-time therapy. You can't reverse the effects of the drug, and you really want to make sure that it works. So what we do is we pick patients that have perhaps more advanced disease. They have maybe some vision loss, they have more changes at the back of the eye, and therefore there is always some risk, which we hope to have understood pretty well and characterized pretty well in the preclinical studies in animals, which the FDA has evaluated in that IND and felt, to be low risk enough to go into humans. But we still want to go into patients that have a reasonable amount of the disease to test if it's safe. So what we do is we identify as many patients as we can with the disease in the pre-screening study. We take really good quality images and assessments that we would want to use to evaluate them later during the study. And we can basically line them up for different parts of the study. And then from there, we can move on to the clinical study itself. So that's the phase 1/2. The phase 1 portion of the study is where we test different doses. So although it's a single dose, there can be different concentrations. They can be a little bit stronger each time. So we start with the low dose. The low dose is a level that we choose where we expect to see some benefit, but is the lowest where we feel it's safe. We will see some benefit, and that's where we want to go first with our patients. And we'll try one patient, and if all goes well, we can dose a few more patients, and then we take all of the data from those patients. We're looking at safety. So, did the patient have any adverse effects? Did they tolerate it well over a long time? Did they see any benefit from it? And we take all of that information. We discuss it with a panel of experts in the disease who are independent to the company, and they help us to give an independent assessment of if they think it's safe. And if they say, yes, great, everything's going well, you can move up to the next higher dose, we can move up to the next dose level. So we've passed the low dose level, that's great. Now we have to move up to the mid dose. And we go through the same process. We test it in one patient. If all goes well, we can test it in a few more. We collect that data package, and we go to this independent committee of experts. And then with their endorsement, we can move up to a higher level. And if we get through all of that, and we get all the way to the highest level, and we see that the highest dose is doing really well, that may be the dose that we take forward to the second part, the phase 2 portion of the study. And that's where we want to find more patients to test the drug in, to reassure ourselves that it is safe in a wider population of patients, because as you know, people can all respond differently to the same thing. And to also give us a better understanding of how well it's working in this wider population. And so that's the phase 1/2 portion of the study. Now, if we get to the end of that and we find that we have a really strong clinical data package, we can again go back to the FDA, get some feedback from them on that, and start designing out a phase 3 study typically. But again, as Amber mentioned, there are ways to sort of sometimes merge a phase 2 into a phase 3. There are many, many different ways that you can design clinical trials. For rare diseases, you typically try to make it a little bit shorter than a traditional phase 2 going into a phase 3, because you have a limited pool of patients, and those patients are desperately searching for something to help them. So you want to kind of get to the finish line as quickly as you can. And that's where it makes a lot of sense to go back to that pre-screening study and go back to that pool of patients that you've already identified and that you have some really great data on from when you first tested them and they didn't have any treatment, to when you bring them into the study and then treat them, and then you can see the difference. So you can see how the disease was progressing when they had no treatment, and then you can see how their disease progresses, or hopefully doesn't, after they've had the treatment. And the more you can show that difference and the more data points that you can show, the stronger your argument is for showing that hopefully the drug actually has benefit. So that's just a very kind of quick overview of how you progress through the clinical trial development process. And then, if you get to the end of phase 3 and, you've passed all of those hurdles, you're the 15% that made it, then you go and you submit this new drug application. And you can go through, again, fast track processes and accelerated approvals to try to get it to patients as quickly as you can. And then once it's out there and patients are taking it, you can get that real world data in the phase 4 and continue to evaluate and make sure that it's really doing what it's supposed to do, and there are no sort of unexpected long-term effects that are cropping up either. So that's the clinical trial process. Now, going back again to our actual clinical studies. So as I mentioned, we have 2 studies ongoing. We have our pre-screening clinical study, and if you go to clinicaltrials.gov and you type in Stargardt, and then in the alternative section you can type in Ascidian, you'll find both our clinical studies. So we have the pre-screening study and the phase 1/2 clinical study. For the pre-screening study, we're actually able to test bringing patients as young as 5 years old. And if they have a genetic report that they already have that shows they have mutations in the ABCA4 gene, and the specifics are actually on the clinical trial website, and they have some clinical manifestations of the disease, which means they're either starting to lose their vision a little bit, or they've been to see a retina specialist and they have some changes detected at the back of the eye, then you can reach out to Ascidian through the email available on clinicaltrials.gov, or you can go to one of the study sites that's listed in that on the web page, and you can get further evaluation to see if you could enroll in the pre-screening study. So that's a nice way to get into the study. If you have a genetic report but maybe you haven't had your vision tested recently, or you haven't had some retinal images taken recently, that could help us to understand if you would otherwise be eligible. And then, so that's nice. We're able to see pediatric patients and adult patients in the pre-screening portion. And then going to the actual phase 1/2 clinical study itself, that's actually only for adults right now. And that's pretty normal. You want to test it in adults who can consent for themselves. And if we show some really good safety and efficacy data, is it actually working, halting the disease at some of the higher doses, we can potentially bring in pediatric patients at a later stage and test in them as well. And we'll have them pre-identified because we have this pre-screening study and we have all the data, so that's really great for siblings. And so going back to the details of the clinical study, that's active right now. We have 10 sites in the US that are going to be enrolling patients. Some of those sites will also be doing the surgical procedure. And the surgeons that we've selected are really experts at doing this procedure. They've done it many, many times for other clinical studies. So we're very selective about that to ensure safety. And all of the enrolling sites are actually very active in inherited retinal diseases or in retinal studies generally, and they have large populations of Stargardt patients. However, it's always helpful to find more patients that are out there in the community that may also be eligible for the study, because as we continue the development process, we will need more patients to participate. And so in terms of the actual clinical study itself, is for ages 18 and up. There are some visual acuity, so how much vision loss you have, there are ranges for that outlined on clinicaltrials.gov. So you have to have some changes to your vision. You also have to have some degree of change to the back of the eye, so some atrophy in the back of the eye. And in order to find out if you're eligible from that perspective, you would have to go to one of those 10 sites and be evaluated by one of the physicians. And if you're interested in learning more about this study, and if you have a genetic report as well, that's always helpful. You can reach out to the, I think you can reach out to Amber, or you can reach out through the clinicaltrials.gov email that's provided. And we can hopefully bring you in that way and see if you're eligible for the study if you are interested. But we are essentially very much in the thick of activities with the phase 1/2 clinical trial right now, identifying patients, activating all of our sites. And to give you an idea of where the sites are, we have 2 sites in Texas. We have a site in San Francisco and one in Oregon at OHSU. We have the Massachusetts INEA right here in Boston. We also have a site in Cincinnati, Ohio. And we also have 2 sites in Florida, in Gainesville and in Miami. And apologies for missing Michigan, we also have the University of Michigan. So we have 10 sites across the US, all really great retinal specialist centers. And I know that they're all sort of excited to participate. They're looking for patients and really great people to work with as well. And so in terms of actually getting, I think one thing I wanted to touch on was the importance of having a really great team, a really great clinical team that works together with the clinical sites to make these studies happen. There's a lot of back-end activity that goes on before we bring our first patient in. And so it relies on having really good retinal physicians who understand the disease and are excited by the study. It involves incredible site coordinators who honestly make our job so much easier. They talk to you, they talk to the patients they already know, and they can provide more information on the study and sort of coordinate things at each clinical site. And they communicate with our team within the company itself to continue moving the study along. And then, we also work with a whole ecosystem of, a specialist center for looking at the images that are taken, the retinal images that are taken from patients and understanding if the disease progresses. Specialist companies that look at the genetic reports and help us to understand who's eligible and who's not. So this whole sort of ecosystem works together to get the clinical trial up and running. And then we have the 10 clinical trial sites as well. So this is a sort of very busy and also very costly process. And so if we can identify patients quickly and bring them in as soon as we're ready to bring them into the clinical trial and move up to the next stage more quickly, it gives us a greater chance of success at getting all the data we need, passing through that stage of the development process and moving up to the next phase. And so, we always encourage participation at events like this. We love talking to you, and so please come and talk to us after this, and happy to take any questions.
My name is Paul Protectus. I have a 26-year-old son with Stargardt and a defective ACDN-01 gene. He's participated in some studies at the National Eye Institute. A couple of quick questions is, with the AAV, how do you keep the genetic material from migrating to other parts of the eye or throughout the body? And part two is, it seems like what you're shooting for would hopefully restore some of the functionability of the ABCA4 gene, and therefore perhaps halt the progression of the disease. But it doesn't appear that it would likely positively affect the photoreceptors, if I'm correct, and therefore improve or restore vision. Is that correct?
Yeah. So, so the first part was about the migration of the material. So that's it. I'm really glad you brought that up. So for our study, we're doing a subretinal injection procedure, and I think Bob talked about this earlier. What this means is we take the actual drug, and these expert retinal surgeons inject it under the retina, right by the area of atrophy, so the part of the eye that's affected by Stargardt. And so the nice thing about that is, even though it's a surgery, it's not just a pill that you can take or something you can quickly inject in the eye. it does mean that the drug goes right to the photoreceptors and the cells that we really want the drug to target. And because it's sort of enclosed in a pocket between the retina and the other tissues of the eye, it doesn't escape, to spread around the body necessarily. So we're pretty confident from our preclinical studies and from our experience in other similar subretinal injection gene therapy studies that there isn't too much concern about sort of systemic spread or side effects.
Yeah, just to add, so in our preclinical studies, in particular those non-human primate studies, we collected every major organ after these experiments and looked for what's called biodistribution of the drug. And even though we were quite confident based on previous subretinal programs that we would not see accumulation of the drug in other organs, we confirmed that with our product in particular. And the second thing I'll add to this is that, as I mentioned, our drug is editing the RNA that's produced by the cell. So there's an inherent control of that editing, meaning if a cell is not normally expressing ABCA4, there's nothing to edit. Therefore, you don't have risk of changing things in cells that would not normally be expressing that gene. Now, while that's not as a significant concern for ABCA4, there are other genetic disorders where that is very important, where you do not want to, for example, overexpress a protein in a cell that would not normally express that. And again, because the RNA exon editing technology works at the level of pre-messenger RNA, the cell itself controls whether or not it will be edited by that normal expression profile. And then your second question was related to whether or not the way if I could paraphrase it, is whether we could improve vision, right? And so I think you're correct. Right? The goal of this program is to slow progression, hopefully stop progression. But if a photoreceptor has died as part of the course of the disease and the cell is no longer there, this medicine will not regenerate new photoreceptors. So as part of the inclusion criteria in our phase 1/2, we're really trying to understand what's the benefit we can provide to those photoreceptors that are so preserved. And there's evidence that really in the macula, you only need about 10% of your photoreceptors to maintain your vision. And so we do think that there is a nice treatment window in the Stargardt population where there are still photoreceptors there. They may not be functioning properly because of the ABCA4 mutations, but as long as those cells are still there, it gives us hope that we can, at minimum, slow progression and hopefully completely stop the disease in its tracks.
And I will actually add to that. I said that in a phase 1/2 part of the study, we pick patients that have moderately advanced disease because we're going in there for the first time. We want to make sure it's safe. But we're also looking for signs of efficacy. We're trying to see if it does have the ability to stop progression of the disease, slow down or stop, or if there's any kind of improvement, improvements in retinal sensitivity, which may or may not translate directly to an improvement in visual acuity, but may result in an improvement in sense of light intensity or color vision or something else. And so in our phase 1/2 study, we're collecting data on lots of different assessments, which includes contrast sensitivity and low light visual acuity, which is something that people with retinal degenerations often notice first as their vision starts to go. When we move to later stages of the study, we start to modify the type of patients we're looking for, because we're trying to, eventually the goal is to try to treat as many patients as possible. So we may bring in patients that have slightly less severe disease. They have more photoreceptors that are maybe starting to go but haven't completely died yet. And those photoreceptors potentially do have a chance of seeing some improvement or reversal if their environment is corrected. And then I think the goal is for any drug is at some point to be able to reach patients that have absolute minimal disease, but you know that with the genetics they have and the fact that they're showing the first stages of disease, that eventually it will become very severe if you don't treat it. And that's where it's nice to be able to get pediatric patients where, you know, perhaps you have a sibling that has already started to experience the unfortunate effects of the disease, and then you have a sibling who has the same genetics, and you know that you could potentially stop any sort of progression or worsening of disease in them. So in this stage, we're going for the advanced, and we potentially cannot bring back photoreceptors that have completely died, but there will be a point where we can treat patients with absolute minimal disease and give them real hope for the future.
I think there might be a question in the back, and then go ahead.
My name is Adam Robert. I was wondering, what was the process like when you first started researching Stargardt at Ascidian, since when you started in 2020, you were pretty much overlapping with researching COVID-19. Were there any hurdles throughout the process?
No, I don't think the COVID-19, other than the fact that it was difficult for our scientists to sometimes work in the laboratories, but we did everything we could to ensure safety in terms of, some of the, similar regulations in terms of just.
I was curious because it was like the big year for that industry compared to others.
To some degree, I think there have been lessons, in particular related to manufacturing of drugs, that the industry as a whole, not just Ascidian, has benefited from COVID. If you think about how quickly it showed that this industry can manufacture drugs, I think that is one of the outcomes, even though as terrible as the pandemic was from a scientific point of view.
Right, because I know there's a big YouTuber, the blind community, who has Stargardt. Before you researched called the blind right.
Thank you. Yep. Do you want a microphone? I think she's coming.
Yeah. He's probably in his 40s now.
I just want to get your point.
Yes.
First of all, this is a great, now have a real clear understanding of how the whole process works of studies. But the first question was very much close to mine. One of the minority of people who have a late onset Stargardt. When I was a alumni here, like 10, 12 years ago, I had a late onset, which is now pretty much maxed out. So I was very much concerned about the photoreceptors activating. I think that the dimers, I think they call them clusters of what I believe to be metabolic debris or whatever. But now my question is always a little bit. When the photoreceptors are activated, are totally activated, now I'm learning like there's what you say, only 10% of them is actually necessary for functioning. But let's assume that they pretty much are atrophied. They don't function anymore. Can they be restored, or are we now getting into something different, like stem cells?
I'll start with that. I think the key thing is to sort of define, right? So if you think of atrophy, and is the cell gone, which would be full atrophy, and there's ways to measure that based on sort of the autofluorescence. And in that case, a gene therapy or a small molecule or anything, if those cells are gone, then this is not going to restore them. But in many cases, the photoreceptors are still present. They're not just working function. And so there are ways through optical imaging and other tests to assess where you are on that spectrum. And some of that is incorporated within our inclusion criteria related to the current clinical trial.
They're pretty much maxed out now.
Yeah, I'm sorry to hear that. To Bob's point, often with diseases like Stargardt, and actually with Stargardt, you have areas of complete atrophy and you have areas of partial atrophy. So the areas of partial atrophy are sort of like the gray area. And literally the gray area is that part where your vision is starting to deteriorate, but you still have some sort of visual or light response in that area, and there are many ways to image the eye to determine where those areas are. And so, perhaps in your case, the area of complete atrophy where all of the photoreceptor structure has completely deteriorated down to the layer below it, those areas are difficult to recover with things like a gene therapy, but there are always areas on the edge of that complete atrophy, which are essentially the next cells in the tissue that will begin to deteriorate. So that's why your atrophy is progressing, because the edge continues to deteriorate the healthy cells adjacent to it. And so one of the hopes is, when we do this treatment and we do the subretinal injection, we hope that the treatment goes all the way around the area of atrophy and sort of really protects the edge of the atrophy, so that the cells there that may be in partial stages of degeneration have a chance to recover if that's possible.
There's a question in the back. Do you want a microphone? Go ahead.
Hey, I'm David. You mentioned the lipid deposits that accumulate. I think, are those called drusen? Have I got that right? Anyway, those lipid deposits, I wonder if the study protocol is going to be monitoring those and whether they perhaps even diminish. Is there the body has some way of diminishing those? That's a data point that could be collected.
That's a great question. We've definitely talked extensively about that, both within our team and also with the IOD experts that we're talking to, both in the US and internationally, and also with the imaging experts we're working with. So there are two ways to look at it. Those hyperfluorescent flecks, those deposits of lipofuscin and other toxic byproducts. There is, an understanding in the community that when those areas diminish, it sometimes indicates actually negatively that the area underneath is atrophying. But that's in eyes that haven't been treated. So there's no reason for the eye to have any other mechanism to remove those deposits. So when they start to disappear, you believe that the cells underneath that are creating those deposits are failing and stop producing the product, essentially. However, recently there have been talks about whether changes in the way that those deposits look, or the way that they spread out, or indeed if they increase or reduce, has some bearing on either the effectiveness of treatment or the progression of the disease. So that is something that we are actively talking about and will be analyzing, and we are taking all the right sorts of images to be able to do that as well.
I'm wondering what other clinical trials are in progress in the same patient population, and whether in your pre-screening study that database that you're generating is something that would enable patients to enter a different clinical trial or benefit additional clinical trials if they're not a fit for yours or something like that. Is that a shared database?
So that is not a shared database, but you typically have to have a genetic test at minimum, a genetic report available to be brought in even for the pre-screening study. So holding that genetic report, which you own yourself, I think is something, if you find that you're not eligible for our studies, you can certainly go to explore other options as well.
Okay. Yeah. What other trials are ongoing in this space?
So one thing I can say, so right now, the ACDN-01, the Ascidian trial, is the only clinical stage program that is addressing mutations in ABCA4, so trying to correct the underlying cause of the disease. There are some other therapies out there, but none of them are addressing the actual cause of the disease.
You mentioned before that this gene also houses many other diseases on it besides Stargardt's. you are working on that gene in the first 25 weeks of it. Are you looking at other diseases and the reactions to mutating this gene or whatever? Or is it only Stargardt's?
So the actual title of our study is ABCA4 retinopathy. So people who have mutations in the ABCA4 gene in exons 1 to 22 are the patients that we will be looking at. And what happens with mutations is they can range in the severity of the impact that they have. So you can have ones that have a minimal impact on the ABCA4 gene, and therefore the protein that's produced can be fairly functional or somewhat functional, and then it goes all the way to the other end of the spectrum where you have really severe mutations that completely sort of stop the normal ABCA protein from being formed, and then you get really severe variants of the disease. So when I think I must have mentioned that what I'm saying is there are, we know a minimum of, I think, 1,200 mutations that have been identified on the ABCA4 gene. And, any person with Stargardt that you meet might be carrying a different combination of mutations. And that's why you all have you have some similarities in your experience of the disease, but they're pretty different. And so when we do a clinical trial, we do have to be selective in picking out a subset where we can use a uniform set of assessments and follow them and have some expectation of understanding of how they will progress. So at one end of the spectrum, you can get really severe mutations that can burn out the center of the eye, and you get the scotoma, but then they also continue to progress beyond that, and you get much, much more extensive disease. That requires a different set of assessments to be able to monitor over the period of time of a clinical study. And then you will get some people with variants that are so, I won't say so minor, but maybe less severe. So they may have a disease that progresses very slowly or comes on very late in life and only slightly impacts their vision. And they would also not be suitable for the study because if they're not changing a lot, there's not a lot for us to measure. So what we do is we pick the best subset in that population where they have a reasonable amount of disease, is going to progress quickly enough that we can detect a change if we treat them. And if we can prove that it works in them, by natural extrapolation of the way the technology works, we can expect that it would actually work in everybody because we're still correcting all the mutations on that half of the gene.
So what I guess I'm asking is, does this extrapolate onto like macular degeneration?
So it depends on the cause for the macular degeneration. I think in the past, people have believed that often in patients over the age of 55 or 60, where they start to see these kind of atrophic lesions in the retina, it was assumed that it was the more commonly understood version of dry macular degeneration. And there was no genetic testing done because there was no treatment for the disease and there was no reason, it was progressing slowly, so there was nothing to be done. I think now we're getting a better understanding with all the genetic reports, and we can see that there is a subset within the dry AMD, the dry macular degeneration population, that is actually due to very late onset Stargardt. So maybe for those patients, again, genetic testing helps to direct them if they have the ABCA4 gene, and we can do testing to make sure that they don't have any of the genes that commonly cause the other form of dry AMD. They may potentially be candidates for this in the future.
So I think where I'm really going with this question is, since Stargardt is such a very kind of rare and small subset of this, and you explained the millions, if not billions of dollars it takes to do research and investigation and to get to the point where you use a drug, you're not going to make your money back on the Stargardt people. So where are you going with this? And I was also curious, for a company like NPD, who's funding you?
I can. So I think our CFO would disagree. He's not here, obviously. But I think, no, in all reality, I think, yes, of course, you have to understand the commercial outlook of a product. But as an organization that's also developing a platform where we think we can apply RNA exon editing to a multitude of diseases, we believe Stargardt, the unmet medical need is there. It is a rare disease, but there are probably upwards of 40,000 patients in the US, so it's not on the rare side. And so we're quite confident in this having a really great impact on patients, but also potential, a return on what we've put into it. And then the who's funding us? So we've actually had a really fantastic venture company called Apple Tree Partners that has put significant. We've raised 90 million through them. Solely through them, so we're not syndicated across different funds, but Apple Tree has been a fantastic company creator over several decades, and so they've been great backers of Ascidian.
So everything you're doing, so I'm hearing 85% of the time folks try to bring drugs to market. And by the way, thank you very much for trying. As always, thank you. 85% of them don't make it to market. It takes 10 to 15 years to do it. Should we be excited about what AI can do to speed up the study on dosage increases and how much, each of the phases that historically bringing a drug to market has always taken this amount of time through all your stats that you gave us. Is AI exciting? Is that going to bring it and speed it up, light speed it along?
My opinion is I'm cautiously optimistic, meaning I think we're moving towards that. I think there will be some areas where there can be some quick wins, but it is not going to overhaul the drug development process. Aspects of data analysis are really getting streamlined through use of computational tools. We do it in our early design, but we can apply that to, for example, the imaging data that Aaliyah and her team are collecting. And all of that is being read at a single center, and they're using quite sophisticated computational software to evaluate those images in a very consistent manner, and it's quite quick, right? As opposed to, you could imagine, 10 years ago, it's literally a human being looking at every image and scoring it. So there are areas that AI and computational data sciences are definitely improving, as well as aspects of the genetic testing, right? So years ago, it could take months to get a genetic report back from a patient, and when we expedite it, we can do it in about 3 to 4 weeks now, and so there are areas where we're seeing great improvement. Yeah, I think one thing Amber reminded me was this is a good opportunity to, and it gets to a little bit about the question around the different genetic mutations and whether we could go into dry AMD at any point. And so the phase 1/2 is really designed to give us the best chance of showing not just safety, but benefit in patients. And so one thing that I didn't mention in my talk, but as a reminder, we all have 2 copies of the ABCA4 gene. We get one from mom and one from dad. And the phase 1/2 is designed today to ensure that we're correcting both copies of the gene. So we do sequencing, genetic sequencing. It takes us a couple weeks, and we confirm that the ABCA4 mutations that we can correct are on what we call both copies or both alleles. And so that is part of the requirement of the inclusion, having those mutations in exons 1 through 22. And if you or folks are interested in understanding whether or not you meet that criteria, again, reach out to the email on our website or through clinicaltrials.gov, and we can point you into the right direction. But that is one key thing. We're working very hard to ensure that we're correcting both copies that are carrying the disease mutations. And if there's any further questions on that, I'm happy to discuss it, because I do appreciate the complexity of genetics.